So this is happening in the thylakoid membrane of the chloroplast, and in the thylakoid membrane there is going to be photosystem 2, photosystem 1, and ATP synthase. Up here is the stroma, down here is the lumen. We start out with electrons in here, and we have photons, so light energy, that comes in and excites the electron. That excitement splits H2O molecules, releasing both oxygen and hydrogen. So we have our hydrogen. And then we have an electron chain that the electrons travel on. So, and as they're traveling, they are decreasing. First of all, the electron transport chain establishes an energy gradient. So the electron is losing energy, and that energy is being released, and it is pumping the hydrogen into the lumen. Now we'll get to photosystem 1 in a second, but these guys are going to travel this way, and because of osmotic, what's the word? Phosphorylation. So there's a high concentration of hydrogen, and they want to pass through to a lower concentration of hydrogen. So osmotic phosphorylation. They travel through ATP synthase and up into the stroma. So the hydrogen go up here and they turn this, and this puts ADP, and phosphate groups, and it makes ATP. These end up leaving and going into the stroma. Now we're going to talk about photosystem 1.
Okay.
I'll have to do another cut of this, to be honest.
So basically for photosystem 1, we have a different electron, which is hanging out in the thylakoid membrane. It also gets activated by light energy, and it breaks away from its chlorophyll group and is accepted by a different electron acceptor. So basically, this electron acceptor is going to pass this electron to NAD+, as well as hydrogen ions that are hanging out in the stroma. And that is going to create NADPH, which is needed for the Kelvin cycle. Alrighty, so this is the Kelvin cycle, which is the second phase of photosynthesis. It's occurring in the stroma of the chloroplasts, and what we have coming into this, the two inputs, they come from the light-dependent reactions. We have ATP and NADPH. So the first stage of the Kelvin cycle is CO2 fixation. So what's going on here is that we have a 5-carbon molecule called ribulose, and what's happening is that carbon dioxide from the atmosphere is coming in through the stromata into the stroma. And what's going to happen is that the carbon molecule from CO2 is going to join with ribulose and then these two oxygens are going to be, are just going to exit the system. And basically what happens after this is that this ribulose molecule, it gets split through an enzyme called ribisco into 3 carbon molecules called 3PG. So that takes us into the next phase. So the next stage of the Calvin cycle is called CO2 reduction. And what's going to happen is that the 2 molecules of G3P are going to move over. They're each going to receive a phosphate group from ATP, and then ATP is going to become ADP. And then next, NADPH is going to be reduced. And each of these molecules of G3P are going to receive 2 electrons. So we have created an entire new molecule called G3P. So basically, the goal of the Calvin cycle is to get 1 molecule of glucose out of this. And to do that, the Calvin cycle needs to go around 3 times. So within 3 turns of the Calvin cycle, we are going to actually get 12 of these G3Ps, and we only have 2 represented, but that's okay. So there's going to be, these are going to move into the next stage, which is our regeneration of our UBP. So there's going to be 2 of these G3Ps which donate their carbons to be able to make glucose, because glucose is C6H12O6, so it has 6 carbons, which is what we need. And then the rest of the 10 G3Ps are going to go through a complex synthesis to create our UBP again. So now we have a 5-carbon molecule to begin the entire cycle over again.